morning to you all. Myself, I am Dr. Indira, Associate Professor in Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, Andhra Pradesh, India. Along with my colleague Dr. K. Prakash Narayan Reddy, we both are giving a series of lectures related to the course Microbiology. So this is for second year BTech Biotechnology students. So far we covered various topics from second unit, that is major groups of microorganisms. In this we covered diversity and classification, major characteristics used in taxonomy, and the characteristic features of viruses. Today we will see the characteristic features and economic importance of the bacteria. Okay. Before going to this, what are bacteria? We all know about bacteria are microscopic in nature, and these bacteria play an important role in our daily life. Every day we are eating so many food products, bakery products, all consist of bacteria. Okay. And whatever the alcohol, beverages produced by bacteria, and again the biogas produced by bacteria. That means these bacteria play an important role in various fields: food industry, pharmaceutical industry, and beverages, and then leather industry, textile. In each and every field, these bacteria play an important role. Not only the beneficial, these bacteria, they are harmful. They causes the infections. Okay. So in order to understand the role of bacteria in beneficial aspect as well as harmful aspect, we need to understand the structural features of bacteria. What the bacterial cell looks like, what are the structural features, how it replicates. All these aspects we have to learn. So today we will see the characteristic features of bacteria. The objective of this topic here to overview the characteristic features of bacteria and their economic importance. So after gone through this topic, the expected outcome is the characteristic features and economic importance of bacteria in various fields will be known. Okay, we will see all these features as well as importance of the bacteria in various fields. Now see the bacteria, these bacteria are evolved around 3.5 billion years ago. That means these bacteria, they are dominating the earth till so far. Even before the evolution of eukaryotes, these single-celled prokaryotic organisms are evolved first on earth, and these are unicellular in structure. You observe these prokaryotic in nature, and these are the single-celled organisms. And these bacteria we can observe in entire ecosystem on the earth. We can observe in soil, water, and even in our body also we can observe these bacteria. And these bacteria, not only the beneficial effects, these bacteria, they causes the infections in humans, animals and plants. And if you see the bacterial history, these bacteria were first observed by Antony von Leeuwenhoek. He is the father of microbiology. Here in this picture we can observe Antony von Leeuwenhoek. So Antony von Leeuwenhoek is the father of microbiology. He observed different shapes and sizes of the bacteria by using his microscopic lenses. Actually, he is having the hobby of making the lenses. Those lenses he used for observing the water bodies as well as his own samples. And next, this bacteria is a Greek word. It is nothing but small stuff. The bacteria is a small stuff. And later, in the golden age of microbiology, the Robert Koch and Louis Pasteur, these two scientists, they contributed a lot to the microbiology field. They developed the solid media, they developed Petri plates, and they identified the disease causative agent. Till so far, no one identified what is the causative agent for a disease. The Robert Koch and Louis Pasteur, they identified the disease causative agent, that is the microorganism, the bacteria. So they identified Bacillus anthrax disease also. Now we will see the animation.
Welcome to Lummu Math and Science. In this video, I'd like to talk about the general structure of a bacteria cell. A bacteria cell is a prokaryotic cell, which means it does not have a nucleus or membrane-bound organelles. Bacteria are made up of a single cell and reproduce by fission or by forming spores. Many times you will see bacteria cells in colonies. Here are several bacteria cells grouped together. Here's a picture of a bacteria cell under an electron microscope. A bacteria cell has an outer covering called a capsule or a slime layer. Bacteria with a thick outer capsule are called gram-positive, and bacteria with a thin outer covering are gram-negative. In addition, bacteria have a cell wall and a cell membrane which makes up the inner layer of this outer covering. Next, you will find cytoplasm inside a bacteria cell. It is a liquid substance and is the location of many biochemical reactions. You will also find ribosomes inside a bacteria cell. Some are found in the cytoplasm, and other ribosomes are attached to the inner layer of the capsule or the cell membrane. You will also find genetic material inside a bacteria cell. Although it's missing the DNA, you will find most of this genetic material in a nucleode, which is a single strand of DNA that is folded. And you will also find circular strands of DNA called plasmids. In addition, you will find gas vacuoles, and some bacteria have a whip-like tail called a flagellum. And other bacteria have pili, which are small fibers that sprout from the cell surface and help the bacteria attach to certain surfaces. So there we go, the general structure of a bacteria cell.
So we have seen an excellent video describes the structural features of bacteria. Okay, we will go in detail how the bacteria looks like, what are the external features, what are the internal features. Okay, see here, this bacteria, they are prokaryotic cells lacking the nucleus and cell organelles. We see eukaryotic cells, they contain nucleus and cellular organelles. That is the difference between prokaryotes and eukaryotes. And they have cell membrane as well as cell wall made up of peptidoglycan. So this peptidoglycan is very, very important. So whenever any infection is there, we can target this cell wall material, cell membrane material. We can develop the drug molecules. We will see the external as well as internal features of bacteria. See the basic structures. The bacterial cell, the cell wall is there, cell membrane, cytoplasm, nuclear material spores. And the special structures are capsula, capsule, flagella and pili. So this is the structure you can observe, external as well as internal features. See here, this is the cell wall material and this is the cytoplasmic membrane. This entire thing is cytoplasm and the genetic material is embedded inside the cell, in the cytoplasm only. No cellular organelles, no mitochondria, no chloroplast, nothing. So everything embedded in the cytoplasm. And the external features, pili, and this is the flagella structure and this is the capsular material, outer layer. So we will see one by one. If you see the cell wall, so based on the cell wall, we can differentiate the bacteria into two types, gram-positive and gram-negative. So this feature helps for identification of the bacteria and we can place the bacteria into a particular group. Already we seen in the introductory part, that is characteristic features used in taxonomy. If you observe here the cell wall material, this is the gram-positive cell wall and this is the gram-negative cell wall. If you observe the structural features here, the composition of gram-positive and gram-negative, the gram-positive cell consists of peptidoglycan and little amount of teichoic acid. And in this gram-negative cell wall, the peptidoglycan structure is very less. Now see the gram positive and gram negative. Gram positive has more amount of peptidoglycan, teichoic acid, and here gram negative, if you observe, this is made up of lipopolysaccharides, less amount of peptidoglycan structure. So this is the difference between gram positive cell wall and gram negative cell wall. So this feature was developed by Christian Gram. So the technique used is the gram staining. In laboratory also you can learn this technique, and this is very important technique for identifying the bacteria based on the gram positive and gram negative cell wall composition. Positive cell wall has more peptidoglycan layer and gram negative cell wall has more amount of lipopolysaccharides. This is the basic difference between the positive and negative. Here you can observe the peptidoglycan structure, what it contains. The peptidoglycan structure contains NAD and NAM. What are these? NAD means N-acetyl glucosamide and NAM means N-acetyl uramic acid. So this structure is very important for medical significance. If you observe here, the NAD and NAM, this is the peptidoglycan structure made up of these sugar molecules, and this enzyme lysozyme this is the lysozyme, and here you can observe the antibiotic penicillin. These two acts on the cell wall material peptidoglycan. They break the peptidoglycan structure. How they break the peptidoglycan structure? They break the bond between the NAD and NAM. Okay, so this is the enzyme and that is the antibiotic. Whenever these antibiotic penicillin, so this breaks the cell wall material peptidoglycan structure, the pores formation takes place inside the cell. For example, say this is the bacterial cell wall. So the penicillin acts on this and forms the porous structures. So what happens, whatever the cellular content, it's leaked out from the cell. If cellular content leaks out, then the lysis of the bacteria takes place. In this way, the antibiotics acts on the bacterial cell. This is one of the mode of action of antibiotic on the cell wall material. And see the cell membrane. This cell membrane performs various functions. This is the site for biosynthesis of the genetic material, cell wall polymers and lipids. And this is permeable. Permeable means it allows the movement of molecules, ions and solutes from in and out of the cell. Transports the electrons, protons which require for cellular metabolism, and electron transport and phosphorylation. These two processes taking place on the cell membrane, and the cell membrane secretes the enzymes, hydrolytic enzymes. So the proteins whatever synthesized, if it is hydrolytic secretory in nature, those enzymes are secreted from the cell by this cell membrane. And the final function of this cell membrane, it gives the protection and shape and structure to the cell material. And cytoplasm. So cell wall we have seen, cell membrane, and next one is the cytoplasm. So this cytoplasm, the genetic material and the proteins, everything embedded in the cytoplasm. You can observe proteins, nucleic acids, lipid, sugars and water. Everything is present inside the cytoplasm. So these are the various components present in the cytoplasm. The genetic material, plasmids, inclusion bodies, ribosomes and spores. We will see one by one. The genetic material. So this bacteria consists of the genetic material, circular plasmid materials. Those are double-stranded DNA molecules. And these plasmids, they are self-replication. So they contain the genes which encodes various properties. For example, antibiotic resistance and virulence factors. And these are not essential for cell survival. So the genetic material is plasmids, and this entire genetic material is in the form of single chromosome located in the cell that is called nucleoid. So this is the genetic material. And inclusion bodies. These inclusion bodies are nothing but aggregates of various compounds. So here in this picture you can observe, so black colored ones are the inclusion bodies. These are nothing but storing reserves, stores the protein material, all compounds in the form of inclusion bodies. And see the ribosomes. In bacteria, the ribosomes are 70S ribosomes. In eukaryotes, 80S ribosomes. See here, if you observe the bacteria, 70S ribosomes, they consist of two subunits. So this is the 70S type, 30S and 50S. And again this 30S encodes the 16S rRNA, which encodes 21 proteins, and 16S rRNA we have seen in the taxonomic identification, where the 16S rRNA is used for identifying the bacteria, phylogenetic relationship between the organisms. And the 50S encodes 2 units, 23S and 5S rRNA. So among all these units, the 16S rRNA is very essential for identification of the bacteria. Now see the endospores. So endospores are the dormant structures. They reserve the energy and storage of the food material. So they reserve the energy material, and here the endospore location is also very important. Based on the location and number, we can identify, we can differentiate the endospore formers and non-endospore formers. This feature is used for identifying the bacteria. Here you can observe in this picture some endospores. They are located central, subterminal and terminal like this. And see the example Bacillus staurodermophilus. This strain produces the endospores. And another one, Bacillus anthracis. This causes the anthrax disease. So endospores are the structures used for identifying the bacteria. And capsule, this is the external structure, external outer layer. Generally this gram-negative organisms produces this capsule type in order to protect from the host system. And not only this capsule, here in this picture you can observe the surrounding layer is the capsular material. This capsule is helpful for attachment, protection from phagocytic engulfment in the host cells, resistance to drying, and depot the waste products, resolves the waste products and releases the waste products, reservoir for certain nutrients, and not only all these, it protects from the host system or surrounding environment. So these are the features of capsule. And now see the external special features, flagella and pili. So these are the locomotory organelles used for movement of the bacteria. Some bacteria, they are mobile and some are non-mobile. And these flagella consists of the protein that is flagellin, and the arrangement of this flagella varies from one bacteria to another bacteria, and the number also varies. Some bacteria, they consist of single flagella, some consist of more number of flagella, and some flagella they arrange one side, and some flagella they arrange all over the body of the bacteria. So the arrangement and number of flagella is also important for identification of the bacteria. Now see the various shapes of bacteria. In this picture you can observe cocci shape and streptococcus, chain form of the cocci shape, and bunch form of the bacteria, coccus structure, that is called staphylococci, and tetrad form, that is called as sarcina. And see other bacilli, chain form of the bacilli, streptobacilli, and here flagellate type of the rod shaped bacilli, and spore former of the Bacillus species. And here others, Vibrio, Kama type shape, Spirilla and Spirochete. All these are the various shapes of the bacteria. So far have seen different characteristic features. What are the external features, what are the internal features of the bacteria. Now we will see the another aspect, that is the economic importance of the bacteria. These are the various fields of where the bacteria play an important role. We will see one by one. Now first one, production of butanol and propionic acid. That means the industrially alcoholic compounds or industrially organic compounds, solvents, acids are produced by bacteria. Here you can observe the example, butanol is produced by Clostridium astabutyrica and propionic acid is produced by propionic acid bacteria species. And the bacteria produces enzymes, several enzymes. They play a role in leather industry, textile industry and detergent industry. Now whatever the detergents we are using, Ariel and Tide, everything contains the enzymes. What is the role of these enzymes in this detergents means, these enzymes acts on the substrate material. The substrate is nothing but lipids, nothing but stains. If any stain is produced on the cloth material, the cloth gives the color, the food material. So it consists of carbohydrates, lipids and proteins. To remove these stain material, why? Because they contain these biomolecules. So these enzymes, they broke down these molecules and releases the stain material from the cloth. So here another example, you can observe streptokinase. This is the thrombolytic enzyme, dissolves the cloth. Okay, so this streptokinase is produced by Streptococcus species. And another important one, the great discovery by Alexander Fleming, that is antibiotic production, that is penicillin. Not only penicillin, several antibiotics are produced by bacteria. Here you can observe the example, bacillin, polymyxin produced by Bacillus species, and streptomycin, tetracycline, erythromycin, all these are produced by Streptomyces species, belongs to the group Actinomycetes. Now they are in the market for treating various bacterial infections. So these are produced by bacteria and again they act on the another bacteria. That is the beauty of the bacteria. And another application in food production, food and food products, dairy products. Every day we are consuming various food products in our home. Curd, butter, all contains bacteria. What are those bacteria present in the milk and curd? Lactic acid bacteria. They are beneficial to our health. And production of yogurt. So lactobacillus and streptococcus cultures are used as a starter cultures for production of yogurt. And another product, that is vinegar. It is produced from ethyl alcohol by fermentation process. And the bacteria used are Estobacter and Gluconobacter species. And another important aspect is the probiotics. This is the new term, you all really heard this term. Probiotics are nothing but live microorganisms. When consumed into our body, they give beneficial effects, health effects, beneficial health effects. So they provide nutrition, they provide vitamins, they provide enzymes, they provide amino acids. So these are the beneficial bacteria when consumed gives the health benefits to our body. And bacteria in fermented food products. So whatever the pickles we are eating every day, so those pickles are produced by fermentation process. So the bacteria break down the complex organic compounds into simpler molecules. And not only fermented pickles, here you can observe the fermented products, the dosa, idli. So all these are fermented by the bacteria. And another fermented products, kimchi and sauerkraut, wine and kefir. So all these are produced by fermentation process using specific bacteria. So these are the various examples for fermented products now in the market. And another application of this bacteria is the bacteria itself, the entire biomass is used as a protein, nothing but single cell protein. Now protein is the product in the market. This is produced by Methylophilus methylotrophus, and this is the commercialized single cell protein. It consists of 72% of protein. So that protein is used as a feed supplement for animals. And other species, cellulomonas and alkaligens, are also used as a single cell protein. And bacteria role in the soil fertility. Agriculture field also these bacteria play an important role. How they increase the fertility of the soil? They fix the atmospheric nitrogen from the atmosphere and convert this nitrogen into nitrates. So in the form of nitrates only the plants able to capture that nitrogen form. So this nitrogen helpful for fertility of the soil. So if you see here in the air, 78% of the air is occupied by nitrogen, and the bacteria play an important role in the fixation of this nitrogen. Here we can see the symbiotic nitrogen fixers and asymbiotic nitrogen fixers. We can see these fixing organisms in this picture. Here you can observe, this is the atmospheric nitrogen. So the nitrogen fixing bacteria, they fix the atmospheric nitrogen and converts this nitrogen into nitrates, nitrites and nitrates. Okay. Later this nitrates are captured or collected by the roots of the plant, and the minerals are transported from the soil to the plant material, and the plant growth will be increased. So in this way the bacteria, here the nitrifying bacteria which converts the nitrites into nitrates. Here fixing bacteria fix the nitrogen into nitrites, and the nitrifying bacteria converts this nitrites into nitrates. So in this form the plants absorb this nitrogen and they grow themselves. And biofertilizers, this is an important aspect in the agriculture field. The bacteria itself acts as a fertilizer. Nowadays the entire environment of the soil, water bodies, the entire ecosystem is polluted with chemical fertilizers. So these chemical fertilizers enters into the food chain and causes the several infections, even genetical disorders and neurodegenerative diseases, as well as muscular system, all are affected because of this chemical pollutants for pesticides. So to avoid all these, we can use bacteria as a biofertilizer. Here these biofertilizers, they improve the cycling of the nutrients and increases the fertility. Not only fertility, they increase the porosity of the soil and increases the water holding capacity of the soil. And these biofertilizers, they can be used in drought conditions also, where the water content is very low. In those cases, these biofertilizers, they increase the water holding capacity for the plant, and they enhance the seed germination. Not only seed germination, they increase the plant biomass, that is the crop yield, by 20 to 30%. And these biofertilizers, they are cheap and friendly. Why? Because the bacteria we can grow in the small scale in a conical flask, and we can produce the huge biomass of this biofertilizer. So these are economically cheap. And decomposers. Okay. Here whatever the dead and decaying organic matter in the environment, these bacteria, they convert these complex organic compounds into simpler molecules. Simpler molecules means minerals form. So these mineral form is utilized by the plants. So in this way they are removing the dead and decaying organic matter in the environment. So they act as a nature's scavengers. And here these bacteria used as a biocontrol agent. Nowadays we are discussing about Bt cotton, Bt brinjal, all these. That means they are controlling the pathogens. Here Bacillus thuringiensis is one of the example used as a biocontrol agent. And another application is bacteria, particularly the anaerobic bacteria. Here in the diagram, you can observe the gas production, biogas production from anaerobic digestion process, where the anaerobic bacteria, particularly the methanogenic bacteria, produces the biogas. So this is the alternate fuel. Nowadays we can use this energy for our needs. And now the summary of this topic. We have seen the various characteristic features of bacteria, where the bacteria are single-cell prokaryotic structures, consists of cell wall, cell membrane, inside the genetic material, DNA and plasmids, and they lack the cellular organelles, and these bacteria consist of 70S ribosomes. In this, 16S rRNA is used for identifying the bacteria, and these bacteria available in various shapes, cocci, rod, bacillus, and in spiral form. And see the economic importance, where these bacteria play an important role in various fields. We have seen pharmaceutical industry, food industry, dairy industry, agriculture, and scavenger activity. All these aspects, these bacteria play an important role in the entire ecosystem. Here these bacteria, they play an important role in various fields. So they play a role in the nitrogen fixation, acts as a biofertilizers, fixes the nitrogen fixing and increases the soil fertility, so that the crop yield also increase. So not only the characteristic features, we have seen various economical importance of the bacteria in various fields. See these study questions. What are the characteristic features of bacteria? Describe briefly. Enlist the industrially important molecules produced by bacteria, and economic importance of the bacteria in various fields, and write the examples of bacteria used in fermented foods and probiotic products, and draw the structural features of bacteria with a neat sketch. For all these questions, you refer the textbook, Prescott, Brock, and any microbiology textbook, and read all these questions for your exam. Okay, best of luck. These are the various references collected for this development of digital lecture. Thank you all.